Hey everyone, it's Mr. Wolfe here, and in this video I'm going to be going over the process of translation. But to just give a broad overview, translation is going to be the second step in the process of protein synthesis. So what we're doing is we're going from a gene, a DNA genetic message, and we're turning it into a protein. Okay, so how's this going to go down? Well, before translation, we started with the process of transcription. And what happened in transcription is we made an mRNA molecule in the nucleus, and now this mRNA molecule is going to come into the cytoplasm of the cell. Specifically, it's going to go to the ribosome, which is in the cytoplasm of the cell. Okay, so this mRNA molecule is going to kind of insert itself in the ribosome like this, and then what you also have just floating around the cytoplasm are these tRNA molecules. Right, tRNA molecules. And attached to a tRNA molecule is an amino acid. So this is, again, the beginning. We're setting the stage for the process of translation. So how's that going to go down? All right. Well, you'll notice that we have some letters here in our mRNA sequence, and then there's also some letters on our tRNA molecule as well. And also you'll notice is that this sequence of my tRNA molecule matches up perfectly with the sequence of my mRNA molecule, right? A with U, U with A, G with C. All right, so specifically this AUG, this sequence, is going to be called a codon, and that's going to be on my mRNA molecule. All right? A codon is a sequence of three letters that code for one amino acid. And so this codon matched up perfectly with the anticodon of my tRNA molecule. All right. So then what we're going to end up what's going to end up happening is we now have our other sequence of our mRNA molecule, this AGC. And that's going to match up perfectly with this tRNA molecule that's going to come by. Right? A with U, G with C, C with G. Right? And again, the codon's matching with the anticodon. Now that these tRNA molecules have brought my amino acids close to one another, these amino acids are actually going to form a bond together. And then what actually ends up happening, and you guys don't need to know this, is that this tRNA molecule ends up actually just floating away like this, but we still have a bond. Let me fix this. We still have a bond between my two amino acids. Okay. All right. So what's going to happen next? Well, we still have some sequence of our mRNA molecule, the CCC. We still have some tRNA molecules, and let's say that this one came in CCC. It really just want to match up at all, keep on floating in. But then we have this tRNA molecule that matches perfectly with the CCC, right? Codon on my mRNA molecule, anticodon here. And again, same thing's going to happen is now that these tRNA molecules have brought my amino acids close to one another, they're going to form a bond. And in reality, this continues happening. Proteins are thousands of amino acids long. But eventually, when the process is done, is that these tRNA molecules end up leaving. Right? The mRNA molecule goes away, and the protein ends up leaving the ribosome. And so what you're left with is a chain of amino acids, and that's going to give us a protein. So that's how, in translation, we go from starting with an mRNA molecule to a protein, and in DNA, in protein synthesis, starting from DNA and going to a protein.